Okay, welcome everyone. Welcome to the Usher Annual Lecture. We're delighted to be able to bring you Sir Martin Landray talking about lessons from and opportunities after the recovery trial. Martin will be taking the stage at about 2 o'clock in about an hour's time. And before that, we've got an opportunity to have the work of the Edinburgh Clinical Trials Unit showcased, and also to bring you up to date with some COVID-19 research from the Usher Institute in general. So my name is John Norrie. I'm the director of the Edinburgh Clinical Trials Unit and Professor of Medical Statistics here in Edinburgh. And just to deal with some practicalities first, we are recording this couple of hours and it is being live streamed, so just be aware of that. Could I ask all of you to post your questions into the Q&A panel? We will be taking questions and answering questions as we go during the presentations for the ECTU showcase. And also we're doing something using a technology called Mentimeter for live polling. This will all be explained by people that understand this technology better than I. But just to bear that in mind as we go through. So just to give you a flavour of what's coming next, the EC2 showcase, we've got Joyce Thompson, our Chief Operating Officer, will give you some background into what the Edinburgh Clinical Trials Unit is and what it does. And then we've got 5 presentations from various Edinburgh researchers. Dave Newby is going to talk about the Hart trial and the use of routine data. Steph Lewis will be talking about the methodology behind statistical analysis plans. Michael Edelstein will be talking about the CC-BOC suicide prevention or treatment trial. Michael will be followed by Sharon Cameron giving a video on the Bridget emergency contraception study, and we'll finish off with Kev Dallywell giving a brief overview of the DEFINE Phase 2 COVID-19 platform study. We'll have an additional 5 minutes at the end for Q&A, and then Sarah Stock and Anne-Marie Doherty will give a brief presentation on COVID-19 research in Usher. We'll take a break for 5 minutes at that point, so five minutes or more, and then it will get down to the main event with Martin Landray talking about lessons and opportunities after the recovery trial. So I'll now hand over to Joyce Thompson, the Chief Operating Officer of the Edinburgh Clinical Trials Unit. Over to you, Joyce.
Thank you, John. I'm privileged to be able to introduce the 5 ECTU showcase presentations today, and these represent the diversity of our portfolio. We will also feature some polling questions using the technology Mentimeter, as John mentioned, after some of these presentations, and this polling function will be facilitated by Dr. Aggie Tello, who is our Senior Research Development Manager within ECTU. So have your mobile phones ready, because taking part in this technology will require you to scan a QR code, which will be shared later on in the presentation. The Q&A session, as John said, will be held at the end of the showcase. So please feel free to add your questions to the Q&A panel function and start each question with the name of the person it's for. Professor Cameron isn't able to join us in person today, but Professor Norrie will answer on her behalf. Okay, so I'll hand you back.
And does that come back to me now, Joyce?
I think it starts with my presentation now.
Okay, on you go.
Good afternoon, and thank you to the Usher Institute for allowing me to present some information on the Edinburgh Clinical Trials Unit at today's session. I'm Joyce Thompson and have been the Chief Operating Officer at ECTU for the past 12 months. Founded in 2006, our UK CRC registered Clinical Trials Unit is based at Building 9 within the Edinburgh Bioquarter. Under the leadership of Professor John Norrie, our dedicated and highly skilled staff work hard to develop, design and deliver clinical trials that support the university's research objectives and improve human health. We have almost 100 active projects within our portfolio, and we are currently collaborating with our chief investigator community in a further 44 projects that are in various stages of development. To make all of this work, ECTU consists of 8 teams who collaborate closely with internal and external stakeholders to deliver and expand our project portfolio. The research development team has been in place for just over a year now, and they act as the initial point of contact for chief investigators who wish to discuss their research project and progress to the funding application stage. As each project is unique, the research development team conduct the early discussions with researchers to evaluate the specific needs of the project and initiate the processes to prepare the proposal for discussion with ECTU management. In the first phase of the project lifecycle, the proposal is received by the research development team and developed sufficiently to allow an assessment of the proposal against the clinical trial strategy. Proposals adopted by the unit are then developed further, and the needs of the study are fully explored through detailed discussions with researchers, our collaborators, and often external partners. This allows the optimal design to be established and the protocol finalised so that the project can be fully progressed to the funding submission with the best chance of success. Should the application be successful, a dedicated project team is allocated and the project moves to the kick-off stage. A myriad of processes and regulatory activities are then enacted to deliver the project to its conclusion, publication, and close down. Today, we are delighted to share some examples of the fantastic work that we are privileged to be involved in. Thank you for listening, and I hope you enjoy the presentations.
We will now hear from Professor Newby, Personal Chair of the British Heart Foundation, Duke of Edinburgh, Chair of Cardiology within the Centre for Cardiovascular Science, on the Hart trial, which illustrates the power of routinely collected health record data.
Hello, my name is Dave Newby, and I'm going to talk to you today about the Scottish Computer Tomography of the Hart trials, with particular relevance to routinely collected health record data. Now, the first Hart trial we did was to ask the question of whether adding CT coronary angiography on top of standard of care would benefit patients presented to the cardiology department with chest pain. These patients are often referred up with stable chest pain, and a referring physician, primary care physician usually, wanted to know the question of whether the patient had angina due to coronary disease. And so we randomized just over 4,000 people to either standard of care alone or standard of care with CT angiography. We wanted to determine whether this would change the diagnosis of the attending clinician, and subsequently whether that would actually change their investigations and ultimately their treatments. And what we found was that the application of CT did indeed change the diagnosis in 1 in 4. It led to a slightly high diagnosis of coronary disease and actually a slightly lower diagnosis of angina due to coronary disease. It changed investigations, including the cancellations of non-invasive ischemia testing, as well as invasive angiography, although it also triggered some extra investigations in patients who had otherwise unrecognized disease. And ultimately, it led to changes in treatment, both anti-anginal therapy and secondary preventative treatment. But having changed the diagnosis, the investigations and the treatment, obviously the next question is, did that actually affect their outcome? So to address that question, we turn to electronic health records data. Now, the beauty of electronic health records data is that, particularly if you've got a country that has a very well advanced system, that you can actually capture the entirety of the data very easily. And indeed, in Hart, over the 5 years of follow-up, we lost very few patients, in fact, only 66 patients overall, which meant that we had data completeness for over 98% of the trial population, which is pretty impressive compared to the standard, which is usually telephone follow-up or clinic appointments or face-to-face consultations. These 66 patients that were lost were actually just purely due to leaving Scotland, and so they were no longer registered with the Scottish healthcare system, and that often leads to my favourite Scottish joke, which you can see at the bottom of the screen on the left there. Anyway, after 5 years, obviously we wanted to look at myocardial infarction, either fatal or non-fatal, given that we have a test that looks for coronary artery disease. And what we're able to show is that essentially doing a CT scan nearly halved the rates of myocardial infarction over those 5 years. So really powerful data. Now, that trial has been criticised because these are using routinely collected health records data. These endpoints were not adjudicated. But actually, the evidence that clinical trial adjudication committees actually make any difference is pretty slim. In fact, systematic reviews by Cochrane have really showed no difference in ultimate outcomes. And actually, the premise that an endpoint adjudication committee is better than any other method has never been shown. Indeed, here's some data that was shared by Jane Armitage University of Oxford in the ASCEND trial. And when they compared the adjudicated outcomes versus the routinely collected data outcomes, actually, they saw very similar effect size and confidence intervals. Obviously, the overall numbers are slightly different because of the different methods, but it does give credibility that routinely collected health records data are indeed robust. Indeed, there are many differences and approach and outcomes, advantages, disadvantages of using a routinely collected health record data. But one of its major strengths is that it's very efficient. It costs very little in the way of money. Very often, if you had a trial size of 4,000 people, it would run into many millions of pounds to collect follow-up data. Whereas routinely collected data can be done for tens of thousands of pounds through robust systems. On top of that, the people that are making the decision as to whether someone's had an endpoint are actually independent of the trial committee and indeed the trial team. So actually, you get a very independent view of whether a person has or has not met the outcome. So this makes for a very efficient, arguably very independent and very clear method of assessing patients and following up in clinical trials. And indeed, that has led us to now consider doing the Hart 2 trial. In the Hart 1 trial, we did find that people that didn't have angina also benefited from a CT coronary angiogram, probably because of incidental finding of coronary artery disease that had nothing to do with their symptoms. In Hart 2, what we want to do is to take asymptomatic patients. And here, what we want to do is to compare CT with the standard of care, which is a cardiovascular risk score. Now, actually, again, if you turn to Cochrane, the evidence that a cardiovascular risk score has any benefit is minimal. And indeed, we know that risk scores often overtreat a lot of people unnecessarily and actually miss a lot of the events that do subsequently occur. So here, the Hart 2 question is it better to screen for the disease with a CT coronary angiogram or use a risk score? And what we're going to do is randomize 6,000 patients who are middle-aged with one cardiovascular risk score to either the score or to the CT scan and see which has the benefit. And obviously, again, we're going to follow these people up with routinely collected health record data over the 5 years to see if it does indeed make an impact. This is again another demonstration of how you can very quickly, rapidly, and efficiently do these sorts of trials using routinely collected data to see whether it actually makes an impact on outcome. Thank you for your attention.
Thank you so much, Professor Newby, for that really interesting presentation and updating us on the results of your clinical trial, Hart. So the way that the polling questions will work is either you could use the QR code by taking a photo of it on the phone, or you could follow this link and there's a passcode just underneath, and there will be a series of 3 small pop-up questions, one after the 3 presentations. So there's going to be one just after Professor Newby's presentation just now, and 2 more questions after the following 2 presentations. And what we hope to do is show you kind of in a live polling way what the popular opinion is around some of these really common and sometimes a bit controversial topics. So if you would please either use your phone just now to scan the QR code or follow the link that's also put into the chat box. Please let me know perhaps through chat how you're getting on, and I will just have a quick peek to see who can answer the very first question, which I will tell you about in just a moment. I'll just give you a moment to log on to the Mentimeter. Perfect. Okay, people are already popping in their questions. So the question is, the results of clinical trials such as Hart can lead to changes in the routine clinical care of patients. So who should be made aware of important findings from trials such as these? Okay, so I see that questions are popping in. We have about half of our colleagues who are on the call today have answered the questions already. So who's in the front runner? Research communities absolutely should be made aware. Policymakers, very important. We can't enact change without the involvement of policymakers. Of course, the participants who volunteer their health and their time to clinical trials would certainly like to know if something is working and should be implemented into practice. Disease-specific charities are really good at supporting us with disseminating important results. So yes, absolutely, they should be aware. And of course, the general public should also be aware. They would like to know how we're improving care and where public funded dollars are going to improve the health and care of patients. Excellent. Thank you very much. I think so that's the first polling question. If any of you had difficulties getting we will display the QR code in front of each of the polling questions. So if you've missed the opportunity to participate in this one, please do join in on the next one. The QR code will be displayed again on the screen. Yes, clinicians, absolutely. Thank you for that comment, Felicity. Okay, so if we are ready for our next presentation, I'd like to introduce you to Professor Steph Lewis, who's the Professor of Medical Statistics in ECTU, and she will give us a presentation on the guidelines for the content of statistical analysis plans in clinical trials. Thank you.
Hi there, I'm Steph Lewis, and please don't adjust your monitor. I do have pink hair. It was a strange thing that happened at the weekend. Okay, so just let me share my slides. So that should be them. Okay, so I'm going to talk about the guidelines for the content of statistical analysis plans in clinical trials. So the first thing you might ask is, why on earth would you want to bother writing guidelines for such a boring document? Well, there are lots of guidelines out there. So there's the Consort guidelines, which tell you how to report a clinical trial. There's the Spirit guidelines that tell you what you should put in a protocol. But they don't go far enough in the detail. So a statistician, if you say to them, I'm going to do a logistic regression to analyse a particular outcome, a statistician will know that logistic regression could be done in a number of different ways. You can adjust for different things. You can adjust for different things in different ways. And all of these matter. And at the end of the day, we want to make sure that we're pinning the analysis down to something really precise so that no one can tell us, oh, please just publish that p-value, it's slightly more significant. And if you're interested in these sorts of lists, then do go to the Equator Network, where all of these sets of guidelines are published, including the one on statistical analysis plans. So this really all began for me in 2012. In Edinburgh, we were thinking about what should go into a statistical analysis plan, and we wrote down what we thought a list, that list of essential items should be. And then each year, twice a year, a statistician from each of the registered academic trials units in the UK, so these are trials units that have gone through a set of criteria to make sure that they are people who know what they're doing and have experience of what they're doing. So Edinburgh is one of those registered units. So we meet twice a year to discuss the kinds of things that are important to us. And in 2012, we discussed the contents of statistical analysis plans, and we agreed at that we should be doing a piece of work to write down the detail of what should be in analysis plans. So we managed to get funding from the Medical Research Council network of hubs for trials methodology research. Everything I talk about in this seems to have really long acronyms. So we got that in December 2013, and the project started in May 2014. And it was led by Liverpool, by Carol Gamble, who's a real tour de force. So that was brilliant in moving this project along. And one of our team, Ash McCrishan, moved to the University of Liverpool to work on this project. So what did we do? The first thing we did was contact funders and regulators of randomized trials and search the literature to identify existing guidance. And none was found, so that was good. What we needed to do needed to be done. And then we did a survey of current practice across the network of the UK CRC trials units. And this highlighted a great diversity in current practice and confirmed that what we needed to do was write some guidance. So then we did a Delphi survey, and that was sent to a variety of statisticians in academic trials units, guideline authors, pharmaceutical industry statisticians, journal editors, regulators. And at the end of the survey, we then had an expert consensus meeting with 12 people with representatives from across that range. Then we came up with a list of items we thought should be in the guidance, and that was reviewed by trials unit statisticians and the expert panel. And then once we got the full list of 55 items that we ended up with, that was piloted in 5 trials. And so I was involved throughout this process. I was involved in the Delphi survey. I was on that expert panel, and then I was one of the teams that piloted the analysis plans. So I got to see this project in all of its glory and all of its detail. And so we developed some guidance, which is too detailed to go into here, and I'll put the reference up in a minute, but these are the section headings of what we came up with. So then at the end of the project, we were discussing where this might get published. And, you know, in the back of our minds, we were thinking this is a nerdy document for nerdy statisticians about the detail of an analysis plan. Who wants to read that? But lo and behold, it got accepted in JAMA, which was absolutely incredible. So it's been really well viewed. It's been viewed something like 95,000 times and cited over 100 times at the moment, which is fantastic. And has it changed what we did in Edinburgh? Well, not drastically, because it turned out that our 2012 list was pretty good. So we've tweaked that slightly, but we're using it pretty much as is. But it was a great piece of work and really wonderful to be involved with such a great team. Thank you very much.
Thank you so much, Professor Lewis, for this really interesting presentation. I must say, though, statisticians are not nerdy. My statistical colleagues in ECTU are some of the coolest people that I know, and I say that hand to heart. So thank you for all of your hard work on it. And I'm sure that based on this presentation, although I recognize that it was, again, a bit pixelated, some of us will definitely change the way that we attend our statistical analysis plans in research. So if I may guide you back again to the Mentimeter. So if you could please, we could have the QR codes again. Perfect. Thank you so much. And just, I forgot to mention, I apologize the first time, that you don't need to log off. The questions will be put onto the same link that you followed the first time. So if you keep that link open on your phone or on your screen, then you'll be able to see. And I see that the next question has popped right up. So did you have a statistical analysis plan for your most recently published research? I know some of us are not publishers, so we don't publish any research, which I see is quite a few of us, but I am very pleased to see, and I'm sure so is staff, that most of us who are publishing research are indeed doing a statistical analysis plan. This is wonderful. And those 12 or 13 or so, please get in touch with us, and Steph will help you with your statistical analysis plan for your next research. I'm sorry, Steph, I volunteered you. Okie dokie. So let's see, how are we getting on? Does anybody, if you have any trouble linking up to the Mentimeter, just pop into the chat and we'll be able to help you get on if you've had any problems to link in. But that looks like wonderful news. So most of you are indeed doing your statistical analysis plans, and quite a few of us are not publishing any research at the moment, so that's okay. And a small handful didn't include it in their most recent research. So that's really, really interesting. Excellent. Thank you so very much.
Hi everyone, I'm Sharon Cameron, and thanks for having me today to present the results of the Bridget Trial, which was published in The Lancet in November. I'm presenting on behalf of the Bridget team, and of course Bridget was about bridging from emergency contraception to regular contraception in community pharmacies. So emergency contraception works by delaying ovulation, but of course it's really important that a woman starts an effective method of contraception straight away if she wants to avoid an unattended pregnancy. And that's the problem, because most women will get their emergency contraception from a pharmacy, but pharmacists generally cannot provide a regular method of contraception. Women need to get an appointment at a clinic, usually a GP, but sometimes a sexual and reproductive health clinic. And in the wait to get that appointment, that places them at risk of an unintended pregnancy. So some years ago, we wondered, well, what if the pharmacy could provide a temporary method of contraception, a bridging supply, until they could get an appointment with their regular provider and access their preferred ongoing method. And what about the progesterone-only pill, the POP, or the mini pill as it's sometimes referred to? What about that as a method of bridging contraception? Because it is extremely safe with very few contraindications, wouldn't that be a good option? And so that gave birth to the Bridget Trial. So Bridget took place in 3 parts of the UK, Edinburgh, London and Dundee, and it was a pragmatic cluster randomised crossover trial. It involved 29 pharmacies and over 600 women who were presenting and who received emergency contraception in the form of oral levonorgestrel. Bridget was designed to have an intervention which was a combination intervention. It was a combination of giving women 3 months' supply of a progesterone-only pill containing desogestrel, plus a rapid access card that they were told they could present to the local participating sexual and reproductive health clinic, and they would be seen as a walk-in and provided with ongoing contraception. The control was standard care, so this is usually when the pharmacist gives standard advice about where they can access ongoing methods of contraception. The study took place between December 2017 and June 2019, and the aim was to determine if this intervention resulted in greater use of an effective method of contraception than just emergency contraception and advice alone. This is the design, the pragmatic cluster crossover study. So the clusters were the pharmacies, they were randomised, and the order in which they delivered either the control or the intervention was randomised. And following that, there was a washout period during which recruitment ceased, and then they recommenced, having switched over to intervention if they started off with control or vice versa. The participants were women who were 16 years or older, who were not using hormonal contraception when they presented for emergency contraception, and who were not on any medications that were known to interact with the progesterone-only pill. And they were followed up at 4 months by a research nurse, by telephone or for an online survey if they preferred. And they were asked questions about their current method of contraception, which, remember, at 4 months would have been 1 month after their supply of the progesterone-only pill had run out if they were in the intervention arm. And they were also asked, if they were in the intervention arm, if they had used their rapid access card, and all women were asked about serious adverse events. So the primary outcome was use of effective contraception at 4 months, and by effective contraception we meant a hormonal method or an intrauterine method, not condoms. So there were 636 women, average age 23, we followed up 64% of them, and we found that there was a statistically significantly higher use of an effective method of contraception at 4 months in the intervention compared to the control. So 58.4% intervention, 40.5% control with the confidence intervals shown, and overall approximately 20% greater use of an effective method of contraception in the intervention group. And that is clinically meaningful. And that difference remains significant even when it was adjusted for a whole range of factors, including age, ethnicity, birth, etc., etc. What methods were they using at 4 months? Well, in the intervention group, the commonest method was the progesterone-only pill, and in the control group, the commonest method was condoms, followed by combined hormonal methods of contraception. There were some other important outcomes of Bridget, notably that repeat use of emergency contraception within 4 months of recruitment was significantly lower in the intervention group compared to the control group. 10% compared to 18% of women needed to use emergency contraception again. The rapid access card, which was in the intervention group, was used by very few women and was really not used at all rapidly, with only 2 women using it to get an appointment at a sexual and reproductive health clinic within a month. And actually, when we went on to look at the databases of the sexual and reproductive health clinics, we found that actually similar proportions of women from intervention and control groups had attended the local sexual and reproductive health clinic for supplies. So suggesting that this rapid access card had not made a meaningful contribution to the success of the intervention. And there were no serious adverse events. So our interpretation from Bridget was that provision of a bridging supply of the progesterone-only pill from the pharmacy with emergency contraception is safe and effective and results in a clinically meaningful increase in use of effective contraception, as well as reducing the need for further emergency contraception. And so we interpret this that if this is implemented, then this increased uptake of effective contraception should lead to fewer unintended pregnancies after emergency contraception. And I'm really pleased to say that actually this is currently being implemented now throughout Scotland. So a huge thank you to the Bridget team, the patient public involvement, the Edinburgh Clinical Trials Unit, and also Chart from Aberdeen, the pharmacists, the participants, our funder, the NIHR, and of course the Lancet for our lovely publication. Thanks very much. Bye.
Perfect. Thank you so much, Professor Cameron, for that really insightful presentation. We don't have a polling question for after that presentation. The next polling question will be after Professor Edelstein's presentation. Okay, so may I introduce you to Professor Michael Edelstein, Personal Chair of Clinical Toxicology, affiliated with the Center for Cardiovascular Science and Center for Pesticide Suicide Prevention. And his talk today is titled Preventing Deaths from Pesticide Self-Poisoning, the CCBOC RCT. Thank you.
Hi, everybody. Thank you very much for inviting me to give this talk and staying to hear it. Hopefully it will not be pixelated. This is a talk about a clinical trial being carried out in Bangladesh with the Toxicology Society of Bangladesh, and here are my co-investigators. So when we think about suicide and suicide risk, what we must understand is that risk period is often quite transient. Many of us have moments of suicidal ideation which might last a few seconds or a few moments. Some people, it lasts hours and days, and they're clearly at much higher risk. But also we think many acts of self-harm are impulsive, particularly self-poisoning. So those impulses will be there, but last a short period of time. And if you can survive that period of time, then people will get on with their life. So we look about self-poisoning, about the case fatality for those different forms. So paracetamol, which is the most common form of self-poisoning here in the UK and in many parts of high-income countries, the case fatality is much less than 0.5%. So for every 200 people who grab a paracetamol and take it in self-harm, less than one will die. If we take some pesticides like Atabron's example here on the left, less than 1% of people will take that because it's very insect-specific. Unfortunately, there are other pesticides like, for example, monoclonal here, when at least 25% of people who ingest these pesticides will die. And actually the reason why people do it will be exactly the same whether they're taking paracetamol or monoclonal. It's what's available at the time. So therefore, the item available will determine whether people live or die. Now, organophosphorus pesticides produce a cholinergic crisis. They inhibit your acetylcholinesterase and cause massive overstimulation of your cholinergic synapses. Now, to improve the care, we can think in terms of various levels. We can improve the medical care, which is working with the people who have already poisoned themselves. Or we can work about administrative control, try to change how communities use pesticides. We can also do engineering controls or public health controls in terms of banning these most toxic pesticides. And clearly that's the most effective ways. But unfortunately, agriculturally, pesticides are still going to be used for some years to come. A few years ago, from a hospital in Tehran, there was an idea that magnesium sulfate may have a role in reducing deaths from organophosphorus poisoning. Now, magnesium sulfate works by blocking these calcium channels you can see here on the pre-terminal synapse, and also at the interface between the two synapses here. And what it will do is reduce the amount of acetylcholine released. It'll also work postsynaptically to reduce the effects of the acetylcholine overstimulation on the muscles and allow the neuromuscular junction to work. This was a small, not really randomized controlled trial, but it looked like it would work. We did work in our pig model here in Edinburgh, which showed also that it might be successful. So we currently have a randomized controlled trial going on in Bangladesh. The 4 red dots are the 3 centers we're actually going at the moment, and the Rajshahi here, which will start in the next couple of days. So this is a pragmatic, unblinded randomized controlled trial. We're comparing magnesium sulfate used in the dose for preeclampsia. So this is a high dose, 2 grams per hour for up to 2 days, compared to nimodipine at the dose that's used for subarachnoid hemorrhage, and can also be given intravenously to unconscious patients, and comparing that with simple saline placebo. We're planning to get 3,200 patients over 4 to 6 sites. And the primary outcome is death, because clearly that's the thing that most communities care most about. Our secondary outcomes looking at intensive care use. One thing we're trying to do is make sure we recruit as few patients who don't need recruiting as possible. When patients come into hospital, they often don't know what they've taken. They simply grabbed a bottle. So we do a bedside cholinesterase check to make sure the patients show clear effect of the acetylcholinesterase inhibition, and therefore we're recruiting patients only requiring treatment. So we started the study in December 2020, massively slowed down by COVID. Here's our team in Chattogram, and this is the 100 patient celebratory cake. We have lots of cakes. And here's our patient, one of our patients in the randomized controlled trial being treated with an infusion here. I think this is going to be the magnesium sulfate arm. So we were delayed for about 6 months to a year getting going. We finally got going in December. Unfortunately, recruitment's quite low. And one of the reasons for that is transfers of patients just don't happen very much anymore. We were planning that patients would be transferred in from multiple small hospitals all around the major hospitals where we're working, and that seems to be happening much less. But you can see we've now got 3 hospitals recruiting. We've now got a steady increase in rate. We need to add 2 more to start to begin to get to where we said we would be. Unfortunately, we're currently at best, we're going to get 2,500 patients, around the 3,500 patients in the time zone we said. So trying to sort out those solutions with the funder and with the Cohort is going to be interesting. Also, one of the things I like at the moment is a WhatsApp, something I've never had before in the randomized controlled trials I've done in Sri Lanka, where we get messages coming in minute by minute showing patients being recruited across the studies. So what we have here, the randomized controlled trial going on in Bangladesh, is the first opportunity to get a new effective therapy for organophosphate carbamate poisoning in 50 years. We know there's at least 100,000 people dying every year from organophosphorus insecticide overdose. So potentially we may be able to save many tens of thousands of lives worldwide as a result of this study. Thank you very much for your time.
Thank you, Professor Edelstein. That was a fantastic presentation and indeed a really important study amidst a global pandemic. So just like CCBOC, a lot of our clinical trials and indeed clinical trials worldwide have had experienced delays because of COVID-19. So our next polling question, if we wouldn't mind popping it up, is around the COVID delays. So who should fund COVID-19 related delays in research? So governments, funding agencies, higher education institutions, or philanthropists. I vote for the philanthropists. But of course, we all have to share the burden, don't we? Okay, so answers are popping in. Sorry, just to remind you to go back to the Menti. If you're having trouble connecting, please let us know through the chat. I apologize, I skipped through the QR code, but it's available in the chat box, the link again, if you have lost it or closed it by accident, and we'll also share again the code if that's okay. There we go. Okay, so most of us think governments should contribute, and of course the funding agencies. And indeed, I think this is the landscape that we are seeing. A lot of the funding agencies have been very understanding and generous in supporting delays encountered by important research studies, such as this one. Yes, the government absolutely is also picking up the tab. Universities, I'm not sure how much support we're getting from higher education institutions at the moment, but certainly they are also contributing, and philanthropists. Yes, Bill Gates, JP Moulton, yes, please continue to support us and help us do these really important studies. Excellent. Okay, well, thank you very much. So I think governments and funding agencies, for the most part, are who we look to for support in these very difficult times. And indeed, they are the ones who have stepped most up to the plate, haven't they, to continue research activity across the board. Okay, well, thank you so very much for participating in these polling questions, and thank you for all the presenters so far. If I may pass now over to Joyce to introduce the next speaker.
Thank you, Aggie, and thank you very much for facilitating the Mentimeter polling. It was very interesting. So now it gives me a great pleasure to hand over to Professor Kev Dhaliwal, who's Personal Chair of Molecular Imaging and Healthcare and a Consultant Physician in Respiratory Medicine for the Centre for Inflammation Research. And he will discuss with us the Define Phase 2 COVID-19 Platform Project. Thank you.
Well, thank you very much. Yeah, thanks for the opportunity to speak. I'm going to describe a trial platform that we set up last year based in Edinburgh, but with multiple sites open. So why Define? So Define was set up to define pharmacokinetics and pharmacodynamics and mechanistic surrogates of drugs working in very small cohorts of patients for COVID-19. And we wanted to prioritise experimental medicine response to COVID-19 as part of a wider network within the UK. But we were doing this leveraging expertise from within the Centre of Information Research, University of Edinburgh, and the wider NHS Lothian colleagues. And the key thing is assets were coming through as knowledge was emerging. So it's a randomised, controlled, open label, parallel group platform experimental study, really at the interface of phase 1b and 2, and there were multiple cohorts. I'm going to describe cohort 2b, which is patients who are hospitalised with confirmed COVID on PCR and had new changes on the X-ray or CT, which showed they had pneumonitis evolving. We recruited the first patient in 2020, and the first arms are complete in 2b. Importantly, as with all studies, it required a broad church of people being involved, especially a unification of the clinical and lab sciences to hand. So the pathway for Define also is important for Predefine, which is bringing assets forward. So we, as we're learning things, are now bringing forward other drugs, and I'll describe some of these drugs. A drug called inhaled Nephamista, drugs that target a cell called the macrophage, drugs that target the monocyte. These are key cells that we now know are playing a role in inflammation, and other antibodies and other targets. We've now completed the IV Nephamista, we've completed a drug called TD139, and we're now starting a virus-specific T-cell. And this is a picture of Edinburgh Bioquarter, and on the right-hand side, members of our team from the Emerge, and also our nursing team and clinical team that were involved in the Define platform. So one of the drugs that we chose to take on was a drug called Nephamista, which really got quite high coverage early on, being a drug that already existed in Japan and Asia, but really had the ability to block SARS-CoV-2 entering into the host. But it had been used in Japan and South Asia as an anti-inflammatory, and most importantly as an anticoagulant. So it displayed polypharmacology, and had been used for 20 to 30 years within South Asia, given intravenously to patients. So it seemed like a plausible asset to take forward. It was also pasted quite high up on the priority list across various countries across the world. So it's administered intravenously, and again, it had been shown it had huge potency against COVID-19 and had been working in murine models very effectively and being an anti-inflammatory. The other drug that we took forward was an inhaled drug called TD139, which blocked fibrogenesis and scarring, and also been in trials for other lung disorders. Hypothesis being that inhaled intravenous were safe and were good and could be taken forward in future trials. We did a lot of daily visits on these small numbers of patients. This slide shows a variety of tests that we took, but it was an intense study, experimental medicine study, to understand if the drugs were working. We had very clear readouts from the study. Nephamista showed more adverse events with significant increase in creatinine, had extremely poor pharmacokinetics, it rapidly degraded in the blood with metabolites accumulating, was not anticoagulant, had no antiviral effect, had no biomarker signal. This despite Nephamista being in phase 2 and phase 3 trials across the world currently. We showed in these 20 patients that it really isn't ideal to take forward in its current formulation. Significantly, TD139 inhaled drug had excellent pharmacokinetics with inhalational delivery. It was safe, and we were able to show that when it was delivered via the inhaled route, it was actually hitting its target in the peripheral blood. And that brings us back to really where we are with the platform, that we've completed some of the phase 1b/2a, but we're bringing forward other drugs and assets forward into this platform to take forward. So just a big thank you to lots of people that were involved. The huge clinical team and multiple multidisciplinary partners in the labs, and also our biological colleagues in the Centre for Information Research, and the funders LifeArc and the drug companies and university collaborators. Thank you very much.
Thanks, thanks, Kev. So we're running a little bit behind time, but we'll pass on the embedded Q&A session and go straight to Sarah Stock and Anne-Marie Doherty, who are going to talk about wider COVID-19 research within the Usher Institute. Sarah or Anne-Marie, are you there?
Hi, John. Hi, everybody. Yes, thank you very much. I think Anne-Marie is here as well. My name's Sarah Stock. I'm a consultant obstetrician and Wellcome Trust Clinical Research Fellow at the Usher Institute. And I'm going to be discussing with Anne-Marie some of our observational studies of COVID-19. Can you see my slides all right?
Yes, we can, Sarah.
Great, thank you.
Yes.
Great. So here we have a huge variety of studies that have been carried out around COVID-19. Some of these are systematic reviews, some of these are trials, but many of these are observational studies reliant on real-world data analysis or surveillance. We're going to focus today on 3 of these studies, ISARIC, which Anne-Marie is going to now tell you about, and then I'm going to give you a little overview of EVE2 and COPS. So I'm going to hand over to Anne-Marie just now.
Thank you, Sarah. So it's a great privilege to be here on behalf of the ISARIC 4C UK investigators. So ISARIC is the International Severe Acute Respiratory Infection Consortium, and it was set up in the wake of the influenza pandemic back in 2009-2010, when it was realised that it was difficult to get a study through setup in time for it to be able to recruit the first patients. So it's been a sleeping protocol since 2012, which meant that we were in the perfect position in order to recruit the first patients when they came into hospital in January 2020. And since then, we've recruited around 250,000 patients, which accounts for 45% of the UK patients. It's a collaboration across the UK. Next slide, thank you. And we've had 2 main outputs. Firstly, we've had a dynamic report, which has reported to SAGE. So this is updated every day, and SAGE have had access to this. And it gives a near real-time idea of what's happening to patients. So which patients are being admitted to hospital, what happens to them whilst they're in hospital, and then what their outcomes are. Thanks, Sarah. And then in addition to this, we've had a number of our SAGE reports, which have translated into high-impact publications. So this is the first one, which came out in April last year, looking at the first 20,000 patients recruited to ISARIC in the UK. And we were able to look at the different demographics and risk factors for mortality. A couple of months later, we were able to look at the paediatric population, and then we were able to develop a risk score looking at in-hospital mortality. We've also been able to look at the complications that people undergo in hospital and its association with in-hospital mortality, the changes in in-hospital mortality over the first wave, and also vaccinations. So looking at which patients still come into hospital having had their vaccinations, and if there are any groups which are at risk for that. Next slide, thank you. In addition to these outputs, I think something that I'm very proud of is this data analysis platform, which we've been able to build, where we've been able to integrate both prospective and routine healthcare data sets. So on the prospective side, we've had the clinical data analysis from not only from ISARIC, but also from Genomic, from FOSP UK, from UK CIC, as well as the more the different analysis, such as the immunology and the genomics, which have needed a flexible compute system. We've then been able to integrate this with the routine healthcare data, which has enabled us to have a fuller data set and also to follow these patients up for longer. Next slide, please. So I think there are 3 key messages to take from ISARIC for why it's been able to look at these patients and be in this strong position. The first is that it was prepared for this. So it didn't just come into being in January last year. It's been a funded sleeping protocol since 2012, meaning that the ethics and all the approvals were in place and the sites were all on board. I think one of our other strengths has been this ability for near real-time reporting and to be able to provide policymakers and government with information that's not available in the routine healthcare data sets. So information about patients' symptoms of COVID before they come into hospital, how sick they are when they come in, and what happens to them whilst they're in hospital, whether they go to ICU, whether they need mechanical ventilation, what treatments and other interventions they receive. And thirdly, I think going forwards, this building of the analysis platform with both the flexible compute and our standard sort of safe haven areas, and then sharing amongst other prospective studies and sharing with researchers further afield so that we can really get the right experts to look at the data that we've acquired. Thank you very much, Sarah.
Thanks, Anne-Marie. So I'm going to pick up on some of those key themes and talk about another sleeper study. EVE was a sleeper study set up in the swine flu pandemic in 2009, and EVE 2, led by the Centre Director Aziz Sheikh, has been very successful in rapidly leveraging the infrastructure and approvals of EVE to provide new data on the pandemic. And it's contributing to policy decisions in Scotland, UK and further afield, as well as a number of high impact publications. The first national level study to show that first doses of COVID-19 vaccines were effective at reducing hospitalisation, continued tracking and monitoring of vaccine safety and effectiveness, including looking at multimorbidity, and directly contributing to, as I say, to policies worldwide. Within EVE, though, we discovered it didn't cover all populations. For example, pregnant women, the infrastructure really was not there to be looking at whole population pregnancy data. And this is one of the things that we set up, therefore, through the COP study, COVID-19 and Pregnancy Study. There's a number of reasons for that. Pregnancy records are generated at the end of pregnancy, generally not in real time, so we couldn't identify pregnant women who were pregnant rather than who had been pregnant. And also data is siloed in different bits of, in different record systems for early pregnancy, later pregnancy and perinatal and newborn periods. So what we've done within COPS is we've brought this data together along with data on COVID infection and vaccination. So we can look at the effects across the whole of pregnancy, including early pregnancy outcomes and terminations of pregnancy. And what we have done with this is are doing monthly releases to Public Health Scotland, influencing vaccine policy, and most recently our recognition that pregnant women we know may be vulnerable to viral infections, and we are seeing indeed that in COVID-19 pregnant women are a vulnerable group to severe disease and critical care admission. Unfortunately, vaccine coverage remains low in this group, and this is really important public health messaging that we're bringing forward. So as of October, COPS had a total of 140,000 pregnancies, nearly, data in it. And our messaging from this is really the importance of team science and collaboration, shared learning within the methods, and the need to consider the whole population, including pregnant women and children. Pregnant women are often purposefully excluded from trials, and this can lead to real problems with messaging and public confidence in the safety of treatments. And I think this leads really nicely on to our next talk about recovery trial, because it's been a real trailblazer in the way that it has been from the beginning included pregnant women, so that we know data on treatments for COVID-19 can apply to this important population. Thank you very much.
Many thanks, Sarah. You had your tea break unexpectedly early, so we're now going to go straight into the main event. Martin Landray's talk. We are running a little bit behind time, but rest assured we will have plenty of time at the end of Martin's talk for discussion, and we are on course to finish at 3 o'clock. So I don't think Martin needs any introduction, and in the interest of time I'll keep this brief. Martin is the Professor of Medicine and Epidemiology at the Nuffield Department of Population Health at the University of Oxford. He completed his medical training in Birmingham, and his research investigates determinants of common diseases through large, efficient, randomized trials. Martin's studies have recruited well over 100,000 participants and have led to regulatory drug approvals, influenced clinical guidelines, and changed prioritizing practice to the benefit of patients. Martin's going to talk about the recovery trial, which went from first draft protocol to first patient enrolled in 9 days and quickly began to establish what did and didn't work, and crucially immediately influencing clinical practice worldwide. This year, Martin was elected a Fellow of the Academy of Medical Sciences and, in addition, knighted. He also leads the Good Clinical Trials Collaborative, which is all about the development and implementation of guidelines for randomized trials. So, fantastic honour to hand over to you, Martin. We're all very much looking forward to the next half hour of telling us about recovery.
Well, thank you very much. Thank you for having me. And if we have my next slide, please. And again. So I'm really going to deal with this talk in sort of 3 parts, if you like, a short introduction of the things I was doing and thinking about prior to COVID, a section, perhaps a longer section on the recovery trial itself and what we learned during the course of that, and then perhaps finish off with a where we go next and what we need to do to make sure we actually do not only learn the lessons, but apply the lessons. And so I think the sort of common theme which you'll get throughout this is really that reliable evidence from clinical trials is absolutely critical to making medical decisions. I'm a medical doctor. I trained as a medical doctor. And the question was always, do I prescribe a treatment? Don't I prescribe a treatment? How do I know it works? And that requires randomized evidence from clinical trials. And too often, those decisions are made in the absence of reliable evidence, either because the trials were never done, or they were done, designed badly, conducted badly, analyzed badly, reported badly. And that is harmful to individual patients and public health. So actually trials are the way in which one delivers better care. Next slide. But trials have been in trouble. If one looks across the landscape, particularly around common disease, the cost and complexity has massively escalated. There is no other walk of life wherein, with all the advances of technology and communications systems, the costs have increased rather than dramatically reduced. So 2 recent trials of PCSK9 antibodies, those are drugs to lower cholesterol given by injection, cost well over $1 billion each. You can't afford to do many of those. So many possible and exciting treatments simply don't get developed, or get developed very, very small niche populations. 85% of commercial trials fail to recruit on time and to target. And there's by and large a temptation to abandon randomization and they instead look at the lure of observational methods, which are not robust in the context of trying to tell the difference between treatments that might have some effect from those that have none. So there's distorted the treatment priorities. Early decisions are made to kill off drugs which might be useful. We've often moved away from preventive and long-term treatments for common diseases and focus on very expensive drugs for rare diseases. There've been huge advances in that space of expensive drugs for rare diseases, and that's important. But if you look around at what's causing the NHS and every health system around the world to crumble, if you look around at the top 10 causes of global burden disease, it's the common stuff that is common, it's the common stuff that has an impact. Next slide, please. And to illustrate this, here is the information on the top 10 drugs being sold in the United States in 2000 and 2015. Between those 2 dates, 15 years, there was a 2.5-fold increase in the revenue. But the number of patients for whom those drugs were relevant had reduced 7.5-fold. So you have nearly a 20-fold difference in the increase in the cost per patient. And why? Because instead of developing treatments for indigestion, heart disease, diabetes, and depression, things have moved to really quite rare conditions. We have to fix this problem if we want a future health, let alone a future health system. Next slide, please. And so in February last year, Rory Collins, Louise Bowman, Richard Pito, and I wrote this paper in the New England Journal of Medicine, a provocative title, The Magic of Randomization versus the Myth of Real-World Evidence. And what I'm going to go on and articulate for you is what I think we need is the magic of randomization in the real world. And I'll show you how we have approached that. I'd like you to read this paper, but if you don't get a chance to, I'll summarize it on the next slide, please. And it's simply that, first of all, one needs smart design and delivery, integrate with the routine healthcare and data systems that are out there, supported by proportionate trial regulations and guidance for the benefit of patients and public health. And I'll come back to that later on. So that's all I want to say about introduction. If we just move on now, and I take you to the 9th of March, 2020. And at that point, these were the headlines. You can see deaths in Italy soar as 16 million put on lockdown. We weren't imagining that there would be lockdown. I should say that actually it was on the 8th of March was the last flight I took before the lockdown, and that was coming back from Murrayfield, having watched the rugby there. So really, you know, the UK was not thinking about lockdown at that point. What we were thinking about was whether there was enough toilet roll. Don't be so shellfish. Now it's rationing. Of course, things changed very rapidly after that. Next slide, please. And it was on the 9th of March that I bumped into Jeremy Farrar on my way to the Wellcome Trust to work on the morning on the number 18 bus. And in the 10-minute conversation we had between Marylebone and the Wellcome Trust's main building in Euston Square, we were exchanging stories, particularly about the horrors that were happening in northern Italy. And Jeremy said to me, look, the tsunami is coming. It will hit the UK hospitals in 2 weeks. We're going to be in the same situation. We have to have a trial up and running by then. We agreed that if we didn't have a trial up and running by then, it would be too late. And so, as John has highlighted, 9 days later, 10 days later, we randomized the first patient. Next slide, please. But what were we facing? We were facing a situation which none of us had seen before. And so we could only imagine what the situation might look like, but it turns out our imagination was pretty spot on. An overstretched health service where we didn't know if we had enough beds, enough staff, enough ventilators, huge time pressures and personal stress for those frontline medical staff, very large numbers of unwell, anxious, often elderly, and always alone patients. Very difficult circumstances for all concerned. And what we needed was to get randomization in there so that we could understand which treatments worked and which ones did not. Next slide, please. And if one looks at the rationale, well, it was true at the time, it's a little better now, but it was certainly true at the time, that 1 in 4 of all patients who were admitted to hospital with COVID did not survive that hospital admission. For ventilated patients, it was more like 1 in 3. There was huge uncertainty about which treatments might work. There are literally hundreds of candidate drugs, everything from simple treatments like aspirin through to advanced chemotherapy, and likewise from the sane to the downright wacky, including beeswax and snake venom. People have written me the letters. There are many opinions from many sources, some of them medically qualified, some with scientific backgrounds, some with good prior research, and then some of them with none of the above, but very, very high profile. And there was really no reliable data about which treatments worked and which ones didn't. Most things, was opinion-based or laboratory-based. There are a few inconclusive randomized trials. A big trial at that stage was 200 people, and mostly it was uncontrolled case series. And it struck us there was unlikely to be a single big win, but if we could find a few treatments that had a modest effect, that would be phenomenally important in a common disease like COVID. And that requires large-scale randomization. Next slide, please. And so to go forward, we have to look back. And so I was reminded of this paper, which I had read some years before. Why do we need some large, simple randomized trials? And Salim Youssef, Rory Collins, Richard Peto, all great clinical trialists here pictured with Peter Slight, had written this paper back in 1984, not arguing that every trial should be large or simple, but arguing that if we had some large, simple trials, that on the effects on mortality of various widely practicable treatments for common conditions, that could have a massive effect. And it's interesting reading this paper, as I was doing on the 9th of March, that if one closed one's eyes and replaced the words cardiovascular or myocardial infarction with COVID and coronavirus, everything else slotted into place. Next slide, please. And also on my desk was a copy of the so-called ISIS-2 protocol, the second international study of infant survival, a study of aspirin and streptokinase, blood thinner, clotbuster rather, for acute heart attack. And this was actually the trial that first got me interested in trials. It made a phenomenal difference to the field of cardiology and a phenomenal difference to the field of clinical trials for decades after. But that trial was simple in the extreme. There was a one-page case report form. There were very clear outcomes, dead or alive at 5 weeks after admission with an acute heart attack. There's a factorial randomization, aspirin versus not, streptokinase versus not, which of course means that a quarter of the patients get both and a quarter of the patients get neither. And very, very clear results which changed cardiology overnight. But on the front page of that protocol, it said, by far the most important determinant of the success of the trial is the extent to which the responsible physicians and nurses choose to enter their patients. Hence, the extra work must be and is absolutely minimal. And we wrote pretty much the same thing into the recovery protocol in 2020. Next slide, please. So simple eligibility. We didn't know how to treat patients hospitalized with coronavirus, so they were eligible. Important outcome, mortality. If 1 in 4 people are dying, the first question is, how can we reduce that terrible mortality? Randomization, assigning between suitable and available treatments or no treatment, no additional treatment at all. And a one-page case report form, which of course was online because it's 2020, and extensive linkage through to routine NHS data. Next slide, please. So the linkage through to data, the NHS has one logo. Unfortunately, it has multiple organizations and multiple different data sets. And we brought together, I think it was 25 different data sets, covering everything from hospital admission and discharge, mortality, use of intensive care, COVID testing, primary care data, and so on. Brought all that information in with on the basis that in a busy hospital, in the middle of a pandemic, and in fact at any time, frankly, then why ask research nurses and investigators to write things down specially in your case report form because it's a clinical trial, if the information is already sitting somewhere in the NHS? Next slide, please. And so the recovery trial design, as I say, starts simple. Eligible patients admitted to hospital with COVID. There's a randomization, which originally looked like this. So patients could be randomized to 1 of 4 different active treatments. You can read them for yourselves, or to no additional treatment. Everybody, of course, gets the best that the NHS can offer. So they get oxygen if they need it, and they get their diabetes treated if they need it, and so on and so forth. But these are what additional treatments do you get on top of it, determined by randomization, and then tracked for the key outcomes of 28-day mortality, length of hospital stay, and the need for mechanical ventilation. Next slide, please. Over time, we answered those questions, and in a sense, we had to answer more interesting and more difficult questions, and we got confidence that the methods were working. So we then introduced a factorial element, studying colchicine or not, convalescent plasma versus the monoclonal antibodies versus usual care, and aspirin versus not. We've also had an early phase assessment of a drug called dimethyl fumarate, and I haven't put it on the slide, an assessment of a drug called tocilizumab. And as we answered those questions, if you go on to the next slide, if a patient is admitted with COVID today, these are the options that are open to them. The baricitinib is another immunomodulatory drug, another drug to suppress the immune system. The higher dose dexamethasone we're actually only running in the international centres. I'll come back to that later. And empagliflozin, which is a drug that originally was developed to treat diabetes, but turns out to have big effects in heart failure and kidney disease, and may have an effect here. Next slide, please. And COVID can affect anyone, so the trial is open to everyone. The youngest patient is less than 6 months, the oldest over 100 years, about 2/3 are male and 1/3 women, about 18% are from Black, Asian, minority ethnic groups. We have, I think it's 100 pregnant ladies who have taken part. Every single acute hospital in the country has taken part can still take part. So any patient admitted to any hospital in the UK with COVID is eligible for this trial. Within the first 8 weeks, we had 10,000 patients randomized. Within the first 100 days, we had 12,000 patients randomized and the first results. And you can see that, of course, over the summer of 2020, recruitment was flat because we didn't have any COVID. We got much the same in early summer this year and have further recruitment. The peak rate of recruitment was 1,000 patients in 30 hours. Over 10,000 patients were recruited during the month of January this year, and we're now up to 45,000 patients. Next slide, please. So let's turn to results. Well, on the 16th of June last year, we had the first important results, and this was the demonstration that dexamethasone, a cheap, widely available corticosteroid, a drug that's been around for decades, and every medical student can list off the side effects, that drug reduced the risk of dying in these patients by about a third for those who were so sick that they required ventilation, and by about a fifth for those patients who were on oxygen. And those results were clearly different according to the level of severity of the disease. Now, it's worth reflecting that actually a number of academics had written to the MHRA when we first set up this part of the study in March last year, and said, you shouldn't be suppressing the immune system in people fighting COVID. It's dangerous to do so. You should stop randomizing. This trial shouldn't go ahead. It's great to have theory, but it's better to have evidence. And you cannot get evidence if you don't do the trials. This trial has been so important in setting the standard of care all around the world. Next slide. And on the back of that result, this was accepted into UK practice that evening. The chief medical officers from all 4 nations of the UK wrote to every hospital saying that this should now be accepted into practice. It was then adopted by guidelines all around the world. So we announced it at lunchtime. It was policy by tea time. It was saving lives by the weekend, because the results were clear and compelling, because it was a sufficiently large randomized trial. Next slide, please. And subsequently, people have gone on to estimate that this has saved probably up to 20,000 lives over the course of the last year or so in the UK, and perhaps as many as 1 million lives worldwide. I have no idea whether that number is right or not. I don't think anybody else has any idea whether that number is right or not, but it's certainly going to be hundreds of thousands of lives have been saved by a clear result from large-scale, simple randomization. Next slide, please. So I want to switch to a slightly different drug. This is a drug called tocilizumab. Its technical mechanism of action is an interleukin-6 antagonist. To the rest of us, what it does is it picks one part of the immune system and binds to the receptors, and is essentially blocks the action of that one particular part of the immune system. And in a sense, you could say, well, that's going to work better than dexamethasone, which has rather diffuse actions. Other people will say it will work worse because the immune system has so many different pathways. You block one and the others just, if you like, open up and compensate. Now, the reason I show this meta-analysis is not because I want you to look at the detail, but what I want you to appreciate is that these are the types of trials that were being done. A whole series of pretty small trials to very small trials, including several funded by the drug company that developed tocilizumab, which provide no conclusive result. There was one trial, the REMAP-CAP trial, which some of you may be familiar with, which is again a sort of platform trial in intensive care patients, which did get a, if you like, positive result. But when you put all these together, one doesn't know whether REMAP-CAP just got a bit lucky. Maybe REMAP-CAP absolutely got the right answer, that this is a treatment that has to be used very soon after people go into critical care units, or what's going on. And the overall result is inconclusive. So we're left with something of an inconclusive mess, if one likes. and really wishing that there was more information. So we were able to add to that with the recovery trial. Next slide. And what we saw in the recovery trial was that when we randomized 4,000 patients, we could see a significant reduction in all-cause mortality with tocilizumab. If you click the slide, please, and do it once more. And when we want this into the setting of that same meta-analysis, the results now slot into place. There is an overall reduction in the risk of death in the sickest patients of about 13 or 14%, and that again has changed policy in the UK, all around the world, and will doubtless be the subject of licensing decisions over the coming months. Next slide, please. And then our final, if you like, positive result relates to the monoclonal neutralizing antibodies. So everybody's familiar with the picture of the coronavirus these days, because it's on every newspaper and every website, as a sort of round ball with spiky bits sticking out, which are helpfully called the spike protein. Well, the monoclonal neutralizing antibody is actually a combination of two different antibodies which bind to the spike protein and stop it doing its damage, stop it entering cells, but also allow for that virus particle to be cleared more effectively by the immune system. And I want to take you through the slide, the figure on the left, carefully, if you like. First of all, focus on the black lines. So at the top, we have a black line which ends up with a mortality of about 30%. That's the risk of death among those people who are admitted to hospital but have not yet mounted any antibodies of their own. 30% mortality. And that's roughly speaking twice what you see at the bottom black line, which is the mortality in those people who have raised antibodies for their own but have got into hospital. So first of all, having antibodies in advance or in response to the virus and mounting your own is good news, and those patients are at low risk. But secondly, what we saw was that when you then give antibodies in the form of this Regeneron combination, for those people who have not made them for themselves, giving them antibodies is beneficial and reduces the risk of death by about a fifth. Whereas for those people who already have antibodies, giving them more is expensive and ineffective. I guess the analogy is a bit like vitamins. If you don't have vitamins, if you're vitamin deficient, taking vitamins is extraordinarily helpful. If you do have vitamins, taking vitamins is ineffective and costly, but that's about it. Next slide, please. And that again has changed practice all around the world and is changing and has changed the way we treat patients in the NHS. All these benefits are additional to that original dexamethasone. Next slide, please. And what we could see was that sort of stratification of seronegative, that's it, i.e. people who hadn't raised their own antibodies, versus seropositive people who had, whether you're looking at mortality, which is the top part, that's also the figure on the left, the primary outcome, the chances of getting out of hospital, or the need for invasive mechanical ventilators. In each case, giving the antibodies to people who don't have them is good news. Giving them to people who already do have them is no news. Next slide, please. So that's all the good news, if you like, which is that we've identified 3 drugs which reduce mortality in a disease which is common and where originally 1 in 4 patients going to hospital were dying during that hospital stay. But I think almost as important are the 6 treatments now that we have identified have no meaningful benefit. These are each widely recommended, they've been loudly promoted, extensively used, and are fundamentally useless in this context. So hydroxychloroquine, you will remember being a miracle drug. It wasn't just President Trump who was claiming that. It was true of well-respected, at least at the time, professors of microbiology in France. It was true of the president of Brazil. It's true of many people that, in fact, hydroxychloroquine was a miracle treatment. The UK chose to hold on to its stockpile of hydroxychloroquine and make it only available through the trial by randomizing. And it was very wise to do so because it does not reduce mortality in this patient population. Lopinavir, ritonavir is an HIV treatment, much the same story. Hydroxychloroquine and lopinavir were first and second choice in many international guidelines for the treatment for COVID, and neither has any benefit at all. Azithromycin is an antibiotic, which was thought also to have some antiviral and anti-inflammatory properties. Much better to save the antibiotic for treating bacteria, where it is effective, and not for this virus. Convalescent plasma, 500,000 Americans have been given convalescent plasma. They did a study of 100,000 Americans giving them convalescent plasma. That study was a beautiful example of a large, simple study. Unfortunately, they didn't randomize. They said it was too hard to randomize. And yet, that as a consequence, they had no idea whether they were doing the right thing or the wrong thing. And it turns out that actually the convalescent plasma had no meaningful benefit in these patients. Aspirin and colchicine have also had their advocates and have also had good reasons to hope they would work, but they don't. Next slide, please. So where we've got to is we now know that dexamethasone, tocilizumab, and the monoclonals are effective in these patients, but we have 6 others that are not. Next slide, please. So I want to reflect a little bit about what we've learned and what are the key lessons. Well, this is an article, an opinion piece written in the New York Times by Zeke Emanuel. It was titled, certainly not by me, and possibly not by him, as "Where is America's groundbreaking COVID-19 research? The US could learn a lot from Britain." But I think there's some important lessons here. The recovery trial is easy to take part in. That was deliberate. I showed you that from the ISIS-2 protocol. It was quickly approved at the national level and adopted by all hospitals in Britain. So approvals within 9 days, all hospitals in Britain within a matter of a few weeks. The background patient data provided by the National Health Service helped simplify the research processes. It is true that leaders in government healthcare did ensure widespread cooperation, and they also ensured that the trial was seen as part of clinical care, not an added optional extra or burden on the side of clinical care. And the research nurse system that we have by and large across the UK has also been incredibly important in making this happen. Next slide, please. We managed to embed the trial in routine practice. These data are taken actually, it's 1st of October to 30th of November last year as a sort of snapshot. These data are for England only because those are the data I had to hand. But in blue bars, what it shows you is the number of patients admitted during that time period to each hospital. And in orange dots is the proportion of those patients who were then randomized and recruited into the recovery trial. And you can see that on average, that's about 10%. You can also see that that varies substantially. And even some quite busy hospitals, 1 in 4, 1 in 5 patients admitted to hospital with COVID are going into the trial. This is randomization as part of clinical care. This is what a learning healthcare system really looks like. Next slide, please. And this is what it feels like to the clinicians on the ground. So these are quotes all from NHS consultants. So this one, coming into work each day, people would say to me, they've chosen the wrong drugs. I'd say, let's see. I didn't know that this drug, turned out to be dexamethasone, would work. No one knew which drugs would work, but I thought we should help find out. 3 months on from the start of the trial, we have a therapy which is cheap and readily available. Millions could benefit. Not far wrong there. I'm glad we helped contribute 1% of the data. So one district general hospital contributing 1% of the data, more data than most entire clinical trials around the world have contributed and helping to provide an answer. Next clip, please. The recovery trial has inspired many of the more junior doctors in our trust to look again at a career in research and given an opportunity and access to treatment our patients would not otherwise have had. I mean, I like the first part because I'm an academic. More people joining research would be great. I'm actually much more proud of the second part. It's given an opportunity and access to treatment to our patients they would not otherwise have had. Next slide, please. And a similar quote from a third investigator. In the interest of time, let's move on. But patients are the real why and the how when it comes to these clinical trials. So this is from Kimberly. Being given the opportunity to participate in the recovery trial was very humbling. It might be humbling for her. Boy, it's humbling for me knowing just how many people have taken part. knowing that the information they were collecting had a direct impact on the treatment of patients, and signing on was something I did gladly. Next. I was already so ill that I was willing to give anything a go if it might help me recover more quickly. Okay, but I also knew that it would help the researchers studying the coronavirus to work out which treatments actually help people. I'm really glad that the NHS patients can take part in the recovery trial, because otherwise no one would know what treatments work for the people actually suffering from COVID-19. Next slide. It is a miracle how things have progressed in such a short time. A year before I caught COVID, we had only just heard about this disease, but now we have these treatments that can be offered to people like me. If nobody took part in clinical trials such as these, we would still be looking for something that worked against this illness. Next slide. And COVID-19 was such a big unknown, and I knew that clinical trials were the only way we would find out what treatments actually work or not. I really do think that the treatment, they got tocilizumab, made a big difference. Up until then, it was quite scary, as I didn't know if I was going to make it or not. So we have this, this is just examples of quotes from people who are in, extraordinarily difficult and frightening circumstances, and reflecting that contributing to the trial is important for them, but it's important particularly for future patients. And it is fundamental to actually understanding what works and what does not work. Next slide, please. And I think a key part of the recovery trial has been our approach to transparency and communication. Everything I've shown you pretty much is available on the website, always has been. Every copy of every ethics submission, MHRA submission, protocol version, all the results in lay and technical language, the recruitment numbers live, all is available for patients, for staff, for public, for media in the UK and around the world. Next slide. So what next for recovery? Well, we're continuing to evaluate a number of treatments, as I've said. We have in fact expanded internationally and are doing more. We're currently in Nepal, Indonesia, and Vietnam. And we will be shortly extending to influenza, where there is also a dearth of information. I mean, take oseltamivir or Tamiflu as an example. That's a drug that is often recommended for use, but in fact, there's no evidence base that really gives a clear answer. Baloxavir, another antiviral, and indeed dexamethasone once more. In fact, the idea for dexamethasone originally came from the flu setting. We still don't know whether it works in that setting. So that's something that's that will be coming soon. Next slide, please. And so back to my reflections on sort of where I started. This is the paper that I wrote with colleagues back in February of 2020, and I wanted to particularly focus on the lessons that we've learned from that and where we're going. So next slide, please. Click again, please. So the G7 it's been the British presidency of the G7 this year, and of course they had their meeting in Cornwall in June. And as one of those things, the health ministers signed up to a charter. So the G7 also is sort of G7 plus one, because that includes the EU. And among the things on the charter, clinical trials are the primary way to generate actionable evidence. So I think that's the first time I've seen that actually written effectively into government policy around therapeutics. Next slide, please. Many therapeutic trials have been inadequate in size and design and conduct, failing to generate the evidence needed. The FDA, Janet Woodcock, has published a paper saying 95% of trials that are registered on clinicaltrials.gov for treatments for COVID had no hope of ever answering their question. They were either not randomized or were too small or both. Next slide, please. And so the G7 leaders have committed to implementing these principles in their own countries and with partners around the world. And I think it's our role is to hold them to account for that and make sure that that does actually happen. And we don't forget the lessons of the pandemic. Next slide. There was also, as part of that G7 activity, something called the 100 Days Mission to respond to future pandemic threats. And so this is the G7's contribution to looking ahead. And I think we have to remind ourselves that this pandemic is not over. It may feel over in some parts of Britain at the moment. That may or may not last. It's certainly not over if one looks internationally. Next slide, please. And one of the elements of that was around clinical trial regulation. So there was a commitment in that transform the approach to clinical trial regulation, shortening the time to authorize trials. That's the easy bit to focus on, actually. The harder bit is streamlining the requirements and guidelines relating to trial conduct. So this is quote directly from that report. We should refocus regulatory guidance on the fundamental scientific and ethical principles that underpin randomized trials, whilst embracing flexibility in innovation. The GCP guidelines for clinical trials should be revised to focus on what matters for the generation of actionable information, rather than what is easy to check but less relevant, placing an emphasis on principles and purpose rather than process. Next slide, please. And that dovetails rather nicely with the reason I was going on that bus to the Wellcome Trust back on the 9th of March last year. And that was in my role of leading the Good Clinical Trials Collaborative, which is hosted at the Wellcome Trust and supported by Wellcome, the Gates Foundation, the African Academy of Sciences, but includes contributions from probably many on this listening in today, and many around the world from all walks of life and all settings. Next slide, please. And really it focuses on how we get better guidelines for clinical trials, and how we focus on the why, not the who, what, where, or how, emphasize the principles, recognize that many trials pose little or no additional risk to participants compared to normal practice. You know, in the States, patients were getting prescribed hydroxychloroquine or not prescribed hydroxychloroquine. As a consequence of not randomizing, they couldn't work out whether it worked or not. We randomized, we worked it out. It's no more harmful in the context of the randomization than it is in routine care. The only difference is from the trial, you actually learn something. We recognize the strengths of the routine healthcare system and the standards which our hospitals and the individuals who work in them are already held. We don't need to burden them with yet more rules and training requirements. Pharmacists are very good at dealing with dispensing. Doctors know how to assess a patient and prescribe. Nurses know how to assess patients and interact with patients and how to follow protocols, and so on and so forth. And we should allow trialists to determine what is an efficient and effective solution and discourage excessive or defensive practices. And one key lesson, documentation is not the same as quality. Next slide, please. And so as part of that Good Clinical Trials Collaborative work, we've taken an approach to define what good looks like. Well, focused on the science and the ethics, the guidelines should be clear and concise. Anybody can write more. The question is, can you write less? It needs to be inclusively developed, and we have done this through regulators, funders, commercial academic trialists, clinicians, patients, public, and more besides, and needs to be progressive and durable. Not only as new diseases like COVID pop up, new treatments or interventions occur, new technologies arrive, but also how the relationship between patients, clinicians, researchers evolve over time. Next slide. And so I'll finish with what I've learned. In some ways I've learned nothing. Randomized trials are still an essential component of high quality clinical care. You know, if one believes that million lives saved, and it's an estimate, but it's a reasonable estimate, then those million lives would not have been saved if we hadn't had large-scale, simple randomization. Arbitrary use of unproven treatments is damaging to patient care and public health. It's actually making a false promise to patients. In my view, it's a breach of trust to patients. We need to understand which treatments work and which ones don't. That requires randomization. And we've seen that compelling results change practice. But it's all very good saying, well, we need trials, but there's an obligation on those of us who are responsible for trials. They need to be designed to deliver actionable results. They need to be feasible. They need to be inclusive, and they need to be focused on the things that actually matter. And that's going to require leadership, coordination, collaboration, a sense of fairness and transparency. Now, that was absolutely the case for COVID. It's still the case for COVID, but it's also absolutely true for the huge burden of many other common conditions which affect individual patients, their families, the health system, and indeed the economy. Next slide, please. And so my very final slide is to obviously to say thank you to the funders and those who collaborated with us and supported us in one way or another, but particularly to the thousands of doctors, nurses, pharmacists, and other healthcare and research staff at, I think it's now nearly 200 hospitals actually, who have contributed to this study. But most importantly, to now 45,000 patients who've participated in the recovery trial. When one thinks about the pandemic, one looks at the slides I've shown you, much of what we look at is numbers, but behind every number is a real person, a real family, and sadly, too often, a real tragedy. We do the trials so that we can find better treatments for future patients. And we are enormously grateful to those patients who take part in the trial. Without them, this would simply be impossible. Thank you.
Fantastic, Martin. Many, many thanks. Both inspirational in terms of what recovery achieved, but also tremendously thought-provoking against the even bigger challenges ahead. To, how do we make the possibilities of recovery the kind of new normal for doing trials? So, sincere thanks from all of us for both doing recovery, but also for a really inspirational talk. There have been, we're unfortunately very short of time now, but there have been a few questions. One coming in from Anthony. Maybe you could just clarify this, Martin. It's a feature of these platform trials, if we've understood it correctly, that the standard of care is evolving. So, when you say, aspirin was useless, that is, by the time you tested aspirin out, would it be the case that most people would be getting dexamethasone as the new established standard of care?
Yes, I'm careful not to say that these are technically that the treatments are useless. In the sense you can never prove that something's completely useless, but they have no meaningful benefit. Yes, by that state, but we can characterize the patients when they get randomized, and so we know that large numbers of them were already receiving heparin. There were 95% of them were receiving dexamethasone, and so on. In that context, we have the results for aspirin, and there's no meaningful benefit.
Great. So I think that is it's a nuance, but it's quite a challenge in interpreting them, because of course there's different part. The second question we had is about, I think you partially answered it, about launching recovery type trials in low middle income countries, where and I think you answered that by saying you were going out to Nepal and Indonesia, but of course there might be differential access to the established standards of care. So I think you've got to be quite careful with the claims. It is always up against a particular standard of care which is not fixed in stone. There was another question, just a couple of questions more about sorry, it's all gone. I think it was a question from Rod Taylor about was there any collaboration from the pharma industry in recovery?
Good question. No, they provided no money, but they did provide drug. So it's actually I think a really effective partnership. So the recovery trial itself is funded by UKRI and NIHR, so government, if you like, funding. But the companies, and so Roche provided tocilizumab, and Regeneron provided their drug, and the Boehringer Ingelheim provided empagliflozin. In each case, they provided the drug, but they have not provided funding, and they have no influence or ability to influence what we then do and how we evaluate it. So in a sense, think of it as a neutral platform, controlled independent from the companies, to which the company then submits their drug, and we then in return produce an answer, which they may or may not like. My job is to provide the right answer, not the one that people want.
Fantastic. I'm afraid we're going to have to. There are more questions piling in now, but we will answer all of the questions to the best of our ability. And of course, there is an opportunity to get in touch with Martin after the talk. But in the interest of time, I'll now have to hand over to Aziz Sheikh, who's going to give the vote of thanks. Over to you, Aziz.
Great, thanks very much indeed, John. And really, we're coming to an end, to the end of an absolutely fantastic afternoon. So just a few words of thanks. First of all, thank you to everybody for joining. Appreciate it's a busy time and really very grateful that you've been able to spare the time. Secondly, I mean, a number of people have been involved in setting this up, presenting today. So I'd like to thank Dave Newby, Steph Lewis, Michael Edelstein, Sharon Cameron, Kev Dhaliwal, Joyce Thompson, Sarah Stock, Anne-Marie Doherty. Other colleagues have been helping orchestrate things, so Aggie with the Mentimeter, Gina Cranswick, Pamela Sinclair. And then obviously, I'd like to thank our comms team and appreciate that they had a really challenging time, but they kind of really rose to that challenge and got things functioning again. So thanks very much indeed to Susan, Laura and Dawn for pulling various strings behind the scenes. And of course, also to John for chairing. I think finally, Martin, it's been an absolutely fantastic keynote that you've given today, our Usher Annual Lecture. So we're very, very grateful. Brilliant presentation. It's been groundbreaking work and we overuse that word, but this has been, I mean, I think that this is no exaggeration at all. I mean, saved so many lives. So Martin, appreciate you've got so many calls on your time. So thank you so much for sparing the time. For inspiring us and also showing us the way forward, because I think we need so many more of these large-scale, simple trials as you've shown the way. I think finally, I'd just like to thank the number 18 bus because, I mean, if it wasn't for that and that conversation with Jeremy, I mean, who knows where we'd been. But thank you so much to everybody, and in particular, Martin, and really look forward to seeing you in person again soon. Thanks so much, everybody. Goodbye.